EAPCET aspirants. So, here we are with the third session of sexual reproduction of flowering plants. So far we have dealt with all the pre-fertilization events and pollination, and we have completed up to abiotic pollinating agents. Today we are going to start with biotic pollinating agents. Okay. So yes, under biotic pollinating agents, biotic agents of pollination, of course insects, bats, birds, snails, snakes, everything will pollinate, but then majority of them will be insects. And biotic pollination, as it is by animals, this is also addressed as zoophily. Fine, right now? So now we will see what are biotic pollinating agents. Mostly it will be, major biotic pollinating agents will be what? Insects. And most common biotic pollinating agents amongst insects are again like bees, butterflies, flies, beetles, wasps, ants and moths. Unlike the characteristic features of stamen, stigma and plants of abiotic pollinating agents or plants, biotic pollinating agents will have specific characteristic features to attract the insects for pollination. So, here if you see characteristics of insect pollinated flowers. What are the major characteristics of insect pollinated flowers? Here flowers are usually large and clustered in what? Inflorescences. Okay. Here flowers are large and they are clustered in inflorescences. They are brightly colored petals to attract insects. They have brightly colored petals which will attract the insects. And fragrant flowers are there, which fragrant flowers due to volatile compounds which are present in the flower, and nectaries are present. Sticky pollen grains are present. There in the abiotic pollinating agents, pollen grain feature will be what? Non-sticky and light. But here it is sticky pollen grains, so that the pollen grains can adhere to the insect body. And flowers pollinated by flies or beetles, they are often what? Foul smelling. Okay. Flowers which are pollinated by flies and beetles, they are foul smelling. Floral rewards of insects. What are the floral rewards of insects here? Nectar is one floral reward, pollen, and here ovary will be the safe place to lay the eggs by few insects. Special characteristic feature of yucca plant by the moth Pronumba. Okay. Pronumba moth, what does this do is, this will lay eggs in the ovary of the yucca plant. Okay. Parallelly yucca plant will get pollinated. So, it is a mutualistic or symbiotic association between Pronumba moth so here it is there, right? Pronumba moth and yucca plant, this is a symbiotic association. Moth will lay eggs and yucca plant will give shelter. This is an example for obligate mutualism. Moth lays eggs in the ovary and flowers will get pollinated as such. Both will depend on each other for life cycle completion. Okay. Both are dependent on each other for life cycle completion. Okay. So, these are the characteristics of insect pollinated flowers, and what are the floral rewards that are being given for the insect for its pollination is written over here. The insects are awarded with nectar, pollen which is nutritious, and special example is a place to lay eggs by Pronumba moth. Clear, right ma? Clear till here? Any doubts anywhere? Yeah. Next. Here we go with birds, pollination or ornithophily. Common bird pollinators, here you see common bird pollinators, for example sunbirds and hummingbirds. These are the common bird pollinators. Characteristics of bird pollinated flowers. What are the characteristics of bird pollinated flowers? Flowers here are bright red, orange or yellow in color. Okay, they are bright red, orange or yellow in color. They are usually large and tubular. They produce copious nectar and little or no fragrance at all, those which are pollinated by birds. These are strong flower stalks, they have strong flower stalks to withstand what? Birds visit. Okay. For example, best example for bird pollinating agents include bombax and callistemon. Next is bats, that is chiropterophily. Okay. Pollination by bats is what ma? Chiropterophily. And here pollinating agents include fruit bats. What are the characteristics of bat pollinated flowers? Flowers here, they are large and dull flowered. They will open at night and close during daytime. The flowers are nocturnal here. Okay. And they are strong, fermented or fruity odor. They will produce what? Strong, fermented or fruity odor, and they will produce large quantities of nectar and pollen. Okay. Large quantities of nectar and pollen will be produced. Flowers are borne on strong stalks or trunks. Okay. Flowers are borne on strong stalks or trunks. Examples here you see Anthocephalus and Adansonia. These are the best examples for what? Bat pollinating flowers. And then you see beta pollen or nectar robbers. These are the insects which will take the nectar and pollen, leaving the plant unpollinated. Okay. So, those are what? Nectar robbers. Some insects will consume the nectar or pollen, but they will leave the plant unpollinated. Examples here are important, Seema, very important. Examples for pollen robbers include queens of Bombus affinis, which perforate Aquilega spurs and steal the nectar. Okay. Just remember the insect, that is enough. Okay. Bombus affinis. And here these terms are very important. Of course, you know insect pollination is what? Entomophily. Bird pollination is ornithophily. Bats pollination chiropterophily. Snails pollination malacophily. Reptiles it is ophiophily. Clear, right? So, insects entomophily, birds ornithophily, bats chiropterophily, snails malacophily and reptiles is ophiophily. That terms you have to remember beta. Next here is outbreeding devices. So, here we have observed self pollination and cross pollination right in our previous class. So, how in a bisexual flower or monoecious flower, of course, both male and female reproductive structures are there in the same flower, but stigma will be incompatible for the pollen grains which are released by the anther of same flower. That is known as outbreeding devices. Outbreeding devices are operated only in the bisexual flowers, only in the bisexual or monoecious flowers. Okay. Bisexual or monoecious. Here we go with outbreeding devices. First, see, it is dichogamy. Dichogamy is a phenomena in a bisexual flower. Either male reproductive structure will mature prior to the female reproductive structure, or pistil will mature prior to the anther. Okay. That maturation period will be different. That is learnt under what? Dichogamy. So, if at all male reproductive structure is maturing prior to the female reproductive structure, that phenomena is known as protandry. Okay, that is known as what beta? Protandry. First, protandry. Okay, where male reproductive structure will mature prior to female reproductive structure. Okay, examples include sunflower. Next, protogyny, where female reproductive structure will mature prior to male reproductive structure. Here you can remember in this way beta. Pro means prior, andrecium. Pro means prior, gynecium. Prior reproduction of gynecium is learnt under protogyny. So, here it is protandry and protogyny. Example you can take of Aristolochia. So, this is dichogamy. Next is what? Heterostyly. Okay, next is heterostyly. that is the lengths of style in the female reproductive structure and filament in the male reproductive structure will be varying. Okay? Lengths of style in female reproductive structure and filament in male reproductive structure will be varying. So, if you see here, see protandry and protogyny is explained, right, prior maturation, and here this image stands very good for what? Heterostyly. This is the best example for heterostyly, okay, where here you can see unequal length of anther and stigma. Clear, right? Here it is stigma, here it is anther. How the pollination is possible? Not at all possible. Next. Next is self-incompatibility. The stigma will be incompatible to the pollen grains which are released by the anther of same flower. That is self-incompatibility. Dicliny, okay, which is nothing but providing what? Unisexual flowers in a monoecious plant. Okay, this is providing what? Unisexual flowers in bisexual plant. Fine, right? Next, prepotency. Prepotency is also the same thing, that is prior maturation. Next is hercogamy. Hercogamy is also the same thing, okay, wherein anther and stigma will be incompatible. So, these things you have to keep it in your mind, ma. Dichogamy, heterostyly, self-incompatibility, dicliny, prepotency and hercogamy. These comes under what? Outbreeding devices. Next. Now, we will see about pollen pistil interaction, and then first we will see artificial hybridization, and then we will move on to pollen pistil interaction. First you see, ma, artificial hybridization. What is artificial hybridization? Nothing but, see here the male plant with one desirable character and female plant with one desirable character, we get the offsprings of those parents with both the desirable characters. Okay, to implant the both desirable characters in the next generation, artificial hybridization will stand as a major support in inculcating all the desirable characters that we need. So, here if you see plant 1 is a male parent, okay, here plant 1 is a male parent which is having tall and rigid stems. Here we will be collecting pollen grains from such plant. Next, plant 2 is a female parent, the trait will be having what? Large fruit size, and here female gamete is inside what? Carpel. Okay, female gamete is inside the carpel. Offsprings if you see, ma, tall and rigid stamens with large fruit size. Offsprings will have tall and rigid stamens from male parent, and they will also have large fruit size which is taken from the female parent. So, how can we do this? How can we do this? What is the procedure? In a bisexual flower which already had a male reproductive structure and a female reproductive structure, in such bisexual flower, first you have to cut the anthers. You have to cut the anthers. That phenomena is called as emasculation. Emasculation is nothing but what, ma? Removal of anthers. Removal of anthers from a bisexual flower. Okay, you have to cut it off carefully so that the pollen grains should not be dusted on the stigma. And then second step which is followed by emasculation is bagging. Okay, you have to bag that flower. Now, this acts as what? Female flower. Then you have to collect the pollen grains of the desirable character you want. Those pollen grains should be dusted on the female flower like this. So, here if you can see, this is what? Dusting of pollen grains. Dusting of desirable pollen. Okay. and then rebagging again to escape the contamination of the pollen from outside. Okay. So, this is steps you have to remember ma. Many a times the flow chart of artificial hybridization is asked. First is what? Emasculation. Remember all the time, emasculation is done only in the bisexual flower. So, in a bisexual flower, first emasculation will be done, followed by bagging, and this will be a female parent. Then pollination or dusting the desirable pollen on the stigma, fine, right. After that, rebagging again to escape the plant from the contamination of the pollen from outside. So, this will be the sequence of what? Artificial hybridization. First is what? Emasculation, followed by bagging, followed by pollination of the desirable pollen, and then rebagging. Clear, right ma? So, that is the process of what? This is the process of artificial hybridization. Now, we will learn out what will be pollen pistil interaction. How pollen grains landing onto the stigma, what happens? Okay, compatible pollen grains landing onto the stigma, what will happen? Of course, we have learnt after microsporogenesis, and when we are learning about the structure of pollen grain, that 60% of angiosperms will be releasing the pollen grain in two-celled condition. Yes or no? So, that pollen grain, when it is landing on the stigma, what happens is the pollen grain will be already having what? Two cells, right. One is vegetative cell and the other is generative cell. Vegetative cell is large and it has a regular shaped nucleus. Generative cell is small, floating on the vegetative cell, and it has spindle shaped nucleus. And we have learnt anther wall layer. It has two wall layers, with inner wall layer is intine, outer wall layer is exant, right. So, here in the exine you have germ pore regions or apertures where sporopollenin is absent, right ma. So, here from the germ pores or the apertures, the pollen tube will start growing all across the style. Remember what happens? The pollen tube will start growing all across the style. For example, if 2 to 3 pollen grains are landing on the stigma and they are compatible, what happens? The pollen tubes will be growing parallelly in the styles. And at the time of growth of pollen tube, the generative cell will undergo one more mitotic division in the style region itself, in the pollen tube itself. In pollen tube, generative cell will undergo one more mitotic division giving out two male gametes, like this. Okay, it will give out two male gametes. These two male gametes will enter into the ovary, okay, either through porogamy, mesogamy or chelesogamy. We have discussed, right. So, how the pollen grain will enter into ovary, we will learn. Till here it is clear, right ma? How pollen pistil interaction will happen? There will be a long chemical dialogue between the pollen grain and stigma. Okay. So, in vitro also we can germinate the pollen tubes. We can help in the growth. We can look the pollen tube growth in vitro germination. What you have to do is, in a glass slide, take 10% of sugar solution in a glass slide. Just take 10% of, just a second. In a glass slide, take 10% of sugar solution and provide it with boric acid and some minerals like calcium, potassium, magnesium, okay, and dust the pollen grains in the glass slide. Okay. You can see the growth of pollen tube after certain period of time, like this. Okay, the growth of pollen tube you can observe. What is the composition that you have to give for the appearance of pollen tubes in vitro germination is important. Okay, and this is what pollen pistil interaction. After which we will proceed to what? Double fertilization. Before learning double fertilization, you have to know like how the pollen tube is entering the ovary. Okay, we will learn it here. Okay, how pollen tube is entering the ovary. How pollen tube enters ovary. So, here we go ma first. Inverted ovule, and here is the embryo sac, which is 7-celled 8 nucleated condition. This is the chelysal end, and at the micropylar region you have synergids with filiform apparatus and an egg cell, okay, which is egg apparatus over here. Fine. Polar nuclei at the center. This is micropylar region. Fine, right? Micropyle and chelysa. So, here we go with a standard keynote that if at all pollen tube, most of the time pollen tube will come from where? Micropylar region only. Okay. Pollen tube enters the embryo sac or enters the ovule at the micropylar region. This is addressed as porogamy. Fine. This is addressed as what ma? Porogamy. If pollen tube is coming from the chelysal end like this to the ovule, okay, if pollen tube is coming from the chelysal end to the ovule, this is addressed as chelysogamy. Chelysogamy, example is what? Casuarina. Next, pollen tube enters through the integuments like this, through the integuments, okay. It is called as mesogamy, that is through integuments, right ma? Example here includes cucurbids. Clear till here? Porogamy, chelysogamy and mesogamy. Okay. These are the ways how pollen tube can enter into the ovule. Once after entering into the ovule, what happens? Double fertilization is the concept that is very important. Many a times asked a question in your NEET and EAPCET as well. Double fertilization or triple fusion, we will be calling it as triple fusion. Double fertilization or triple fusion. So, here you see pollen grains entering into the embryo sac. What happens? I already told you. The generative cell in the pollen tube itself, it will undergo one more mitotic division, providing out 2 male gametes, right beta? So, here one male gamete will come to the egg cell, one male gamete will come to the egg cell through the synergids, okay, and one synergid and their filiform apparatus will be degenerated here, okay. So, the very first step is what ma? From the synergids, just a second, yeah. So, the very first step is what? Synergids. Synergids, as they are guiding the pollen tube entry into the embryo sac. This is also known as helper cells or cooperative cells. Cooperative cells. Here the synergids, from the synergids the pollen tube will enter into the embryo sac, right, through the filiform apparatus. Here what happens? One synergid and filiform apparatus will degenerate. One synergid and filiform apparatus degenerates. Okay, and one male gamete fertilizes the egg cell, both are haploid, and this provides out diploid zygote. Okay, which is the diploid zygote. This is the product of fertilization. What is the first product of fertilization, beta? Zygote is the product of fertilization. Clear, right? So, here we go. First thing, after the formation of zygote, what happens is second male gamete. Okay, second male gamete will fertilize the polar nuclei which is present at the center. Okay, polar nuclei or central cell, which is diploid. This is diploid and this is haploid. This will provide out triploid primary endospermic nucleus. Okay, primary endospermic nucleus. As two male gametes are fusing or fertilizing here, one with egg cell and one with polar nuclei, this is called as double fertilization, and this is also known as what? Triple fusion. Okay, as such the zygote will be later maturing to what? Embryo. Zygote will be maturing to embryo, beta. Zygote क्या होता? Zygote undergoes sequential mitotic divisions and embryo बन जाता. Fine, right? It will undergo sequential mitotic division and this will form embryo, which we will learn under the concept of embryogenesis. So, under post fertilization events, we will be learning the formation of embryo both in dicots and monocots. Dicot embryo and monocot embryo, and primary endospermic nucleus will mature. It will also undergo mitotic divisions and it will undergo mitosis and it will form what? Endosperm. Mature endosperm. Endosperm is triploid over here. Okay, endosperm is triploid. Triploid endosperm is formed. Fine, right? So, this triploid endosperm, first endosperm development will happen. After endosperm development only embryo development will happen. Fine, right? So, till here everything is clear, no ma? Today what we have learnt? First thing is about biotic pollinating agents, and then about artificial hybridization, pollen pistil interaction, how pollen tube is germinated, how pollen tube is entering into the ovule, and lastly double fertilization. So, at the end of double fertilization, what all are formed, ma? Zygote and primary endospermic nucleus. Zygote matures to embryo and primary endospermic nucleus will mature to endosperm. Okay. So, in our next class, we will be addressing post fertilization events. Post fertilization events. What are the post fertilization events? First is endosperm development. Here we have learnt in double fertilization how endosperm is formed, and one major feature that I have to tell you is after fertilization, post fertilization, the synergids which are present and the antipodals which are present at the chelae end will degenerate. Important statement to be noted, beta. And here in post fertilization event, the first change is what? Endosperm development followed by embryo development, followed by seed formation, and fruit formation. Clear, right? These are the 4 post fertilization events that we are going to deal in our next class. So, endosperm development, first this will happen, after embryo development will happen, but seed formation and fruit formation will take place simultaneously. So, in our next class, after post fertilization events, we will also be solving EAPCET PYQs. Till then, just be prepared with all the concepts that we have dealt with sexual reproduction of flowering plants, and get ready for the next class, EAPCET PYQ practice. Okay? So, you will soon see me with EAPCET PYQs of this chapter. So, be prepared. Thank you.